welcome back. So we were looking at HMQC in the last lecture, how the 2D HMQC works, what is a pulse sequence, how does it look like. We will continue with that today before we move on to the next experiment. So this was a pulse sequence of HMQC which we looked at. So this is, as I said, the information wise it is similar to a 2D HSQC experiment which we have seen. Similar to that, it has 3 different blocks. So the first block is the inept block, which is slightly different from what we saw in HSQC. It does not contain these pulses 180 degrees here, but the idea is similar to HSQC, that you transfer the polarization from proton to carbon using this delay period, which is 1/2 j, and this j is set according to the coupling constant, which we saw that it varies from methyl to aromatics. So based on these coupling values, we choose a particular delay period, and that ensures that the transfer takes place only for one bond. So you have to keep in mind, we are transferring the magnetization from proton to carbon only through one single bond. We are not going for multiple bond transfer, which is a different experiment. So the second part of the experiment consists of this evolution of the carbon chemical shift, which is excited because of this 90 degrees pulse here, and this carbon chemical shift now evolves during this period, and it is decoupled because of the 180 here from the proton. So remember, we have to decouple proton from carbon and carbon from proton both. So this is during the detection period we decouple proton from carbon, but during the indirect period, detection period, which is indirect dimension, we decouple the proton from carbon. Okay. And then the reverse inept block comes, where you transfer back the polarization to the proton, and the proton now starts evolving or getting detected during the T2 period. So that is a chemical shift of proton which evolves or happens during the T2 period, and that is given by cosine omega T2, and cosine omega T1 is for carbon. So this correlates 2 chemical shifts, carbon along T1 with the directly attached proton along T2. So this, if you recall, is the same kind of an information which we obtained from HSQC. So HSQC and HMQC wise, there is the information wise there is no difference, but what I would say is that the difference mainly relies on the sensitivity. So the HMQC, as I said, the 180 degree pulses which are not here make it more sensitive compared to HSQC, because HSQC the 180 degree pulse degrade the intensity, and that is because the 180 pulses are not broadband, and they affect the inversion, that is the 180 degrees are supposed to invert a magnetization. Similarly, 180 degree pulse are supposed to invert for carbon. They are not really uniform over the entire chemical shift range. So because of that, all the chemical shifts of carbon are not equally affected by this 180, and at the edges of the spectrum the sensitivity goes down. HMQC, the problem with that is that it has this resolution problem, because during this period T1, although I have not shown here, there is also a coupling term which will come along with chemical shift, and that if the T1 is very long, that coupling term also becomes dominant, and therefore that is proton proton coupling now, because that is not carbon proton, we have decoupled from carbon, but we have not decoupled 2 protons from each other. So if there is a proton, for example, let me show you here, if there is a proton which is attached to another carbon here, this coupling, which is 3 bond J coupling, is surely active, it is not decoupled. What we have decoupled is proton to carbon. We have decoupled these 2 interactions, but we have not decoupled this interaction. So this coupling, and not only to this proton, there could be another proton this side. So there could be several proton proton couplings which are active, which are actually present or evolving during this T1 period, and that comes into the expression here, which although we are not shown for a simple simplification, and that causes a decrease in the resolution of the spectrum, because the peak starts now splitting because of the J coupling, and that degrades the resolution. So this is one disadvantage of HMQC over HSQC. So therefore, depending on the applications, people use either HMQC or HSQC. So we will see that soon when we come to biomolecules, what type of experiments are carried out. So this is an example spectrum, again as a schematic drawing of a molecule. This is again glutamine, which we saw in the HSQC case, and you can see here again similar to HSQC, we have 3 carbon proton pairs labeled as 1, 2 and 3. So, one pair is 1 CH. So, this CH is coming up here and that is downfield because of this de-shielding effect from these groups. It comes typically around 50 to 60 ppm, between 50 and 60. Now, the second group here pair is CH2. Now, remember CH2, this H2 is not equivalent. This is something you have to keep in mind because this is a chiral carbon. So, there is an asymmetry in the molecule. So, these are not equivalent, but here as a just to show for simplification I have shown it as a single peak, but there are 2 peaks and the chemical shift of these 2 protons will be either very close or it can be quite different depending on the particular structure in the protein. So, that is the number 2, and the third pair, that is CH2 gamma, this is alpha, this is beta and this is gamma. In the gamma case you can see now CH2 again here it is coming slightly downfield compared to 2 because of this de-shielding effect, but this is exactly similar to what we get in HSQC. So, chemical shift wise there is no difference, spectral pattern wise there is no difference. The final difference, that is the coupling which I was mentioning, is not shown here, but it will also affect in the spectrum. So, these are the advantages and disadvantages, or I would say similarities and differences between HSQC and HMQC. So, let us go through this once more, although we have seen this in the previous slides. Number 1 is HSQC gives you one bond, direct one bond, proton to carbon or proton to nitrogen or proton to phosphorus, whichever is X nucleus correlation. Similarly, here we get the same thing in HSQC, in HMQC we get the one bond correlation. So, these are the similarities. Here there is no splitting along the T1 or F1 dimension, that is because of proton to proton coupling, and this is something which happens here in case of HMQC. And this is which is what I told you, that if there is a 3 bond coupling between 2 neighboring protons, you will end up seeing a coupling, and therefore, that will split the peaks into multiplets similar to what we see in 1D, but that will happen along T1. So, that is not we did not see that, but that can happen if you have a very long T1 value. Now, the third one, the advantages, as I said, is very ideal for macromolecules and it is very popularly used in protein NMR. It has a lower sensitivity compared to HSQC, reason being, one is of course the degradation because of this coupling, and the second thing which I mentioned in the last class was that there is a T2 relaxation. When you have multiple quantum coherence, HMQC stands for heteronuclear multiple quantum coherence, we have sensitivity loss because of T2 contribution relaxation from the protons. So, it is ideal for small molecules. It is also okay for proteins, it is not that it cannot be used. In fact, nowadays we have fast NMR experiments in recent years which have been relying directly on HMQC. So, HMQC is also very good for biomolecules, and if you want to do rapid NMR approaches, then HMQC is the way to go. But when you go for 3D NMR experiments, normally we incorporate HSQC into that, and therefore, HSQC becomes more important in such scenarios. So, let me now give you a few examples, actually practical examples of how HSQC is used in protein NMR. Let us look at this protein structure for a very brief period. This is not a detailed structure analysis, just a quick overview. So, HSQC or HMQC can be used for evaluating the extent of protein folding, structure and aggregation. So, this is one of the very important application of HSQC. You suppose you have a protein with you and you want to know whether this protein is folded in 3-dimensional space properly or not. It could be an unfolded protein, it could be intrinsically disordered protein which does not have any structure, or it could be a protein which has got aggregated. So, instead of a monomer you might be seeing a dimer, you might be getting a trimer in your sample. So, how do we identify those things with NMR? So, this is the application where HSQC becomes very useful. So, this is something which all of us know, that proteins consist of sequence, a linear sequence of amino acids. So, this is just an example here. You have several, let us say you have several amino acids in a protein, you have alanine, you could have arginine, this sequence is arbitrary. So, there is a sequence of amino acids and what also we know is that there is a peptide bond between neighboring amino acid. So, this is a simple schematic drawing of a protein structure, linear sequence structure. This is not a 3-dimensional structure, this is just showing the sequence of amino acids. So, you can see here there is a residue number 1, it belongs to the N terminus. Now, with the N terminus of a protein you have NH3, okay, it is not NH, NH comes only in the peptide bond. Okay. So, therefore, the terminal amino acid is having NH3 and that NH exchanges rapidly with the solvent. This exchanges very rapidly with the solvent. So, normally you will not be able to capture this NH correlation, but here you have NH in the peptide bond, and here as written here there are, suppose you have n amino acids, I can write it as 1 first amino acid and n minus 1 and the last is the nth amino acid. Now, this is the terminal COO carboxylate group. So, this again we know this is the last amino acid in the chain. So, it also has NH, but here it has COO minus. So, now if I do an experiment called N15 proton HSQC. So, you can see here this is a very specific experiment where I am correlating the chemical shifts of the nitrogen, it is this nitrogen with this hydrogen. But what is important here is to note is that this nitrogen is now N15, which means I am not looking at the natural abundance N14, I am enriching this molecule. I would have enriched using some methods which you will see later down the line, but assuming that I have isotopically enriched this whole protein with N15, I will have all the nitrogens in the protein as N15, and I can do a experiment N15 proton HSQC or HMQC where I can correlate the chemical shift of each nitrogen here with its corresponding directly attached proton. So, this is shown schematically in this spectrum here, and this is you can see here this is HSQC spectrum, and you can see this correlations what we are seeing here, each of these spots here are peaks and each of these peak actually correspond to one amino acid. So, one peak here has one nitrogen chemical shift on this axis and one hydrogen chemical shift on this axis. So, this is a nitrogen proton pair, and that pairs are basically these you can see in this picture. So, each of the amino acid, because as I shown here these are n amino acids. So, if you have n amino acids you have n such combinations, n such pairs in your protein, and each of this pair now will show up in this case. Okay. So, that is what we will see, but then the N terminus here, this one, this is not actually an amide, it is an amine, and because of these 3 hydrogens NH3 it exchanges rapidly with the solvent, whereas these hydrogens are not so exchangeable, although they can be exchanged, but normally in folded proteins they are involved in hydrogen bonds. So, therefore, they are protected from solvent exchange, so and therefore, the peaks are not so labile, or the protons are not so labile as these protons, these are amines. So, an amide proton we are looking at CONH comes between somewhere between. So, let me write down the chemical shift range. Typically it comes from 90 ppm to about 140 ppm. So, this is what the typical range is. Usually it comes somewhere from 100 ppm to 135. So, there are this specific range will come visit again when we come to the proteins, and here typically it is about 6 to 6.5 ppm in the proton scale to about 10 to 11 ppm. Okay. So, this is typically the range of amide protons. So, we are looking at amide protons here. So, I would also specifically write this is amide protons. So, this is the typical range of chemical shift what we would get in a protein. Okay. So, another thing one has to notice is that these peaks are spread out, they are not together. So, this spreading out of peaks in this region is very crucial for our understanding. So, as I will show you in the next few slides how this spreading out helps us to get some information from of the protein. So, this is how HSQC spectrum of a protein or HMQC N15 proton HSQC or N15 proton HSQC is analyzed. That means, every peak here corresponds to one amino acid, because each amino acid is NH, but the terminal N terminal, not the C, the N terminal residue will not give any peak because of this NH3. And there is one more amino acid which does not give any peak in the spectrum, even if it is not in terminal, even if it is in the center or middle or anywhere, and that is proline, because proline does not have NH, it has only N and there is a cyclic ring. So, NH is absent in a proline. So, when NH is absent, I cannot see any correlation of N with H in that amino acid. So, that is why prolines are also absent in the spectrum. So, we have to keep in mind that suppose you have 100 amino acids, you have to subtract 100 minus the number of prolines and minus 1 because of this first one. So, that many number of peaks you should expect to get. So, let me write it down more clearly here. Suppose number of amino acids, if you have 100 amino acids in your protein, the number of peaks in HSQC equal to 100 minus number of prolines minus number of the first residue 1. Now, this is for backbone. This is backbone. What is a backbone? Backbone meaning this chain here, this line which you see here, this is the backbone of the protein. We are not looking at the side chain. Side chain comes in this R. So, if you recollect, this is the side chain of an amino acid. We will see this as we go along in the protein structure part for those of you who are not aware of how a protein structure looks like. But this R is called the side chain, and this the backbone is C alpha, CO, NH. These are the side backbones. Now, in HSQC you are seeing the backbone amides or what we saw, but you will also see side chain amides. So, let me go back a few slides. Here you see there is a side chain amide here also. This is in case of glutamine. Similarly, if you have asparagine, there also you have CONH2. So, such kind of a amino acid, also this is an amide group. So, it will also appear in the HSQC spectrum. So, the number of peaks are not just simply given by this formula here which I wrote, it is also additional peaks will come, which will be additional peaks are from the side chain. So, let me add this, the side chain peaks. So, which are the amino acid which can give side chain? You have asparagine, glutamine, you can also have arginine, histidine, and so on. So, we will not go into details of how to right now, we will come back to this later, but these are the extra peaks apart from these numbers which you would expect to see. But typically what happens is we know how to identify these. So, that is a way to do that, which I will come back to it in the later part. And once you know that you have identified the side chain, and if you ignore that in your calculation, then what you are left with is only this part. Now, of course, this is the ideal situation. You would expect that for a 100 amino acid, suppose I have 5 prolines. So, 100 minus 6, 94 peaks, but that is not always the case. It is very rare to see 100% of a peaks in HSQC. So, typically for a very good system, I would expect somewhere between 90 to 100, 90 to 95. There will be always a few percentage of peaks which are not observed in at all. And why is that so? That is because they exchange rapidly with the solvent. So, remember this is an labile proton, as I said, although we say it is involved in hydrogen bond etc., but many of the hydrogens which are in the loop regions might be exposed to solvent, and therefore, they might get exchanged with the solvent very rapidly, and because of that exchange they do not appear in the spectrum because of the fast exchange with solvent. So, in such cases, instead of like ideally expected 94 example, for example, let us say I have 5 prolines, I expect 94 peaks, I may not get 94, I may get somewhere around 90 or maybe even 85. So, the missing peaks are really the challenging part in an HSQC or in a protein. In proteins generally we miss quite a number of peaks compared to what we expect from based on these kind of calculations, but then we have to deal with them using some methods. We will see that if possible, if time permits, but this is the challenging part in a protein NMR experiment compared to organic molecules. In a small organic molecules, if you expect 7 or 8 peaks based on its structure, we normally always see those 7 or 8 peaks. Their intensity may vary, but we will definitely not lose them. but in protein the intensity can vary a lot. So, therefore, if this peak is 100 in intensity, the missing peak will be like less than 1. So, it will be like a large difference. So, therefore, it is very easy to miss them or not observe them in the spectrum, even if you record the spectrum with a very high signal to noise. So, even though I would say the signal to noise is very high, even then you can have many peaks which are missing. So, that is the problem with challenge with protein NMR, and that affects structure calculation, that affects the assignment of the protein and so on. But whatever we have with that itself we have to sometimes manage, and we will see how to improve the spectrum quality in case we want to get more and more peaks. So, this is how HSQC N15 becomes a very useful spectrum to start for any new protein. If you have any new protein, you have to first label the protein with N15. How we label them, we will see that in the second part of this course. But once you label a protein with N15, you can record this spectrum, and let me show you now how the information, what kind of information we can get from this. So, as I said, it can be used for estimating or evaluating the extent of protein folding and structure or aggregation. So, this is again same the figure which I showed in the last class. So, now look at these two spectra here. So, you can see this is for some protein which got aggregated or it became multimeric or it basically started degrading, and you can see here I can hardly see the peaks separation here. Okay. So, these are the side chain peaks which I showed you. There are ways to figure out as we will see later, but you can see here I can hardly count the number of peaks in this spectrum. But this was a protein, another protein where it gives a beautiful spectrum here. So, you can see here it is a folded globular protein, whereas this protein is either unfolded, we do not know, or it could be aggregated. Most likely this is an aggregated system. So, you can see here each of the spots here, peaks, are nicely separated, and each one, as I said in the previous slide, it corresponds to one amino acid. So, the goal of protein NMR or NMR community is to try to get this kind of a spectrum. This is, if you count the number of peaks, roughly it is somewhere between a 90 to 100. So, this is a small protein, this is not a very large protein, but even then if you get a spectrum like this, it becomes very easy to work on such systems, and one can get the structure calc of this protein relatively easier. In fact, this type of system here on the left is very difficult to work at. So, normally this type of proteins we do not work with such systems. What is done is to try to improve the sample condition so that we can reach this level. Okay. So, what are the things one can try? One can vary the pH, maybe this at a given pH it has got aggregated. So, one can try a slightly lower or a higher pH. Normally lower pH is preferred in NMR for biomolecular proteins because of the exchange. The exchange of this NH with the solvent depends on the pH, and this is mentioned in varieties of books. We will not go into detail. Typically at lower pH the exchange is lower or lower. So, therefore, if you go to lower pH the peaks may become better, less exchange, so more sensitive, but at the same time very low pH can also result in precipitation of the protein and so on. So, one has to optimize the pH. Salt is another condition, maybe this is aggregating because of low salt. So, if you add little bit of salt, it can prevent the aggregation and it can result in this condition. Sometimes the temperature is a parameter which has to be optimized, because temperature, higher the temperature, protein may start aggregating, and therefore, we may have to go lower in temperature, so on. So, this has to be optimized so that we get a as clean spectrum like this as possible, and if it is not possible to get to this level, what typically is done is that you go back to the laboratory and reclone, means you remove some portion of the protein which may be causing this problem. You can chop the protein into smaller pieces and so on so forth, or you mutate some amino acids in the sequence to make it more less hydrophobic or more hydrophobic depending on what you are looking at, and therefore, that kind of a change in the sequence or protein has to be carried out to get a good looking spectrum like this. So, that therefore, this kind of a process involves time and effort, and that is really a challenge again in proteins which are not amenable to NMR, because this kind of a spectrum is very difficult to work, because I can hardly separate the peaks, and therefore, one has to resort to some more techniques. It can be also possible that this is not an aggregated protein, this could be a monomer, but it could be high molecular weight. Let us say we are working with a very high molecular weight like 50 kilodaltons or 70 kilodaltons. There we have to then use more sophisticated NMR experiments. A simple HSQC which is shown here may not be sufficient, and there we may have to record what is called a TROSY 2D TROSY experiment, which we will see down the line in the second half. There are more advanced experiments which will probably help in improving the resolution when we go from here to here. So, these are the different challenges which one has to face, but you can see that that experiment is already helping us to give a lot of information. It kind of guides us that look, this is not a really great system, this is a great system, can we do something to go from this particular state to this particular state by changing some conditions, etc. So, HSQC therefore N15 proton HSQC is an important experiment in any new protein before we start looking at the structure of the protein. A second use of this experiment is in ligand binding. So, this is again we will cover in the second half of the course, but this is right now I am just giving an example, trying to make see the applications of these experiments. So, you can see for example, let us say I have a protein and have some drug molecule or a ligand to which the protein binds. Now, this is the binding. So, how do I detect that there is a binding? So, by non-NMR techniques there are varieties of method. We can use ITC, isothermal titration calorimetry, or SPR and so on. From NMR how can we detect? So, you can see this is an example of some protein, and here the black color peaks, the peaks which are colored in black are coming from the free protein, free meaning this protein here which is not having the ligand. But the moment the ligand was added to this protein in the NMR tube itself, so we are directly adding the ligand in a specified concentration to the NMR in the NMR tube to the protein, we start seeing changes. You can see now the red color is the new set of peaks. So, the red color peaks are coming from a mixture or the addition of protein and ligand, means it is coming from this here, you can see this diagram. So, it is coming from the protein ligand complex. So, red colors, the black colors are the free protein. So, you can see there are many peaks which are shifted. You can see this peak has moved here from red to black to red, from here black to red and so on. So, each number here which is shown here indicates the amino acid number. So, this is G204, means it is a glycine number 204. There are the sequence is a long sequence, and the 204th amino acid in that protein in that sequence was a glycine. Similarly, here if you look at here serine 276, means the amino acid was number 276 in the chain, and the serine was that amino acid name type in that particular position. So, in this case you can see it has not moved very much, it is in the same position. But look at this here, serine 240 has moved quite a bit. Similarly, here this valine 251, means valine at position 251 has moved substantially. So, by seeing at this movement of peaks, we can actually find out what are the residues which are located at the interface, this line here, the interface of the protein, because those are the one which might be affected because of this binding. We do not expect this binding to affect residues which are far away here. Okay. So, the residues which are at the interface are the one which are expected to be most affected, and those are the one which are moved substantially. So, therefore, this HSQC experiment, by simply titrating or adding the ligand to the protein, we are able to find out which are the amino acids in the protein which have got shifted, which are binding or which are located at the binding interface. This is the binding interface with the ligand. So, therefore, this is a very popular experiment. We use the word chemical shift perturbation, which is used very properly in drug industry, in drug discovery, in drug design, where we try to find out which are, suppose I have added a drug molecule, which are the part of the protein which it interacts with. So, of course, one thing if you notice here, I should know every peak here the amino acid identity, and that is the part of the whole process called sequence specific assignment, which we will go into the second when we do in the second half. So, you have to identify every amino acid here, identify the label with every amino acid. If you look back here I do not know the identity, I just know there are some, let us say 100 peaks in this spectrum, but I do not know each peak corresponds to which amino acid in the protein chain. That information is not available to us, and that comes only from assignment, and assignment itself is a very challenging task, It takes sometimes months to assign a protein and figure out which amino acid corresponds to which peak here. But once you have the assignment, then you have lot of things which can be done, such as this kind of experiment where you can titrate with any ligand and see what are the changes happening. So, these are the applications briefly I have gone through for HSQC and HMQC. In the next part lecture, we will move on to a last experiment in heteronuclear 2D NMR spectroscopy, which is known as HSQC TOC-C, and that experiment actually will show us how to proceed from there to 3D NMR. So, we will see that in the next class how a 2D HSQC TOC-C is made.